So next will be Joshua Jahner from the University of Nevada, Reno. So Josh, if you want to share your screen.
Can everyone see that all right?
Yeah, that was great. So I'll let you even read out the title of your talk if you want to.
Okay. Hi, everyone. My name's Josh Jahner, and I'm a research scientist at the University of Nevada, Reno. And today, my talk title is Characterizing Genetic Differentiation and Hybridization Among California, Rocky Mountain, and Desert Bighorn Sheep in Nevada. And I'm just really excited to be here today to be able to describe my research to you and tell you about all the really cool things that we saw. I want to thank all of the organizers for letting me be a part of this session. I'm going to continue the tradition of showing lots of pie charts on a map, so it feels really appropriate. Before I jump into the things, I want to just thank my collaborators, particularly Mike Cox at the Nevada Department of Wildlife, Mike Buchowski, who's with California Department of Fish and Wildlife, and then my two advisors at University of Nevada, Reno, Marjorie Matoke and Tom Parchman, who've taught me lots of different things over the years. All right, so before we begin, I just want to remind everyone that translocations really are one of the most commonly used and most effective tools for managing wild populations around the globe. Every year, thousands of individuals are translocated from one population to another to either augment populations or restore native wildlife back to their previously existing ranges. And so it's really important that we understand how translocations are affecting these populations. And unfortunately, most translocations have occurred historically without knowing anything about the baseline population genetic parameters in those populations. And this is really important because there are several potential consequences of translocations for these populations. First, translocations can result in either a loss of genetic diversity or sometimes even the gain. And you really have to think carefully about which individuals you're moving if you want to understand what the effects of genetic diversity might be. Also, there can be an erosion of pre-existing population genetic structure. And that's simply just a loss of the correlation between geography and genetic distance. And finally, and perhaps most importantly, translocations can result in a loss of local adaptation. And by local adaptation, I just mean a mismatch of genetics and the environment. And so individuals might not be performing as well, or populations might not be performing as well as they previously were before translocations. And so today I'm going to be thinking a lot about bighorn sheep, naturally. And you've already seen this slide. Most everyone probably has seen this before, even before Sam's talk earlier. But I'll focus in a little bit on Nevada because that's where all my sampling is going to be today. So it's previously thought that historically Nevada had bighorn sheep distributed throughout the entire state. But like many other states in the Western North America, it suffered rampant declines in the late 1800s and early 1900s because of overhunting, overgrazing, and disease. But through efforts by the Nevada Department of Wildlife over the last 50 or 60 years, many different mountain ranges have been restored with bighorn sheep populations, which is really successful. So let's dive into this a little bit more. So this is a map of Nevada, and luckily we have a good description of what the situation for bighorn sheep was in the past, because John Muir spent a lot of time there. And he said of bighorn sheep, throughout the vast region bounded on the east by the Wasatch Mountains and on the west by the Sierra, there are more than 100 subordinate ranges and mountain groups, trending north and south, range beyond range, with summits rising from 8,000 to 12,000 feet above the level of the sea, probably all of which, according to my own observations, is or has been inhabited by bighorn sheep. And so we know that much of Nevada was covered with bighorn sheep. And this map here shows you the distribution of bighorn populations in 1960 before any translocations occurred. And you can see that there was a core group of herds down in the southern portion of the state surrounding Las Vegas. These are all desert individuals remaining. And then there were a few scattered herds remaining in the Great Basin central portion of the state. And overall, this was about 3,000 individuals in the state. Now, translocations have effectively raised the populations in Nevada to more than 12,000 individuals. And NDOW's strategy was guided by previous hypotheses and analyses of stole morphology of bighorn sheep, old stoles. And this led NDOW to translocate Rocky Mountain bighorn into the northeastern portion of the state, California bighorn into the northwestern part of the state, and then desert bighorn through roughly this lower 2/3 or southern 2/3 of the state. It came out later though, Weyhausen and Ramey did a subsequent analyses on the stole morphology data. They had an expanded database, and they found that California and Rocky Mountain bighorn sheep didn't differ in stole morphology, which led them to conclude that perhaps Rocky Mountain and California bighorn sheep aren't genetically different lineages. And so it really begs the question of whether or not we should have these lineages separated into unique designations or not, or whether they should be managed together. And so today I'm going to talk to you about 3 different questions that I wanted to answer using a genetic approach for the bighorns in Nevada. The first one is there genetic structure between these different lineages with a particular focus on California and Rocky Mountain bighorn sheep? Because there is some question of whether or not these are actually different lineages. Secondly, we wanted to ask if there were any herds with hybrid ancestry. We want to know whether or not individuals are moving across the landscape and perhaps exchanging genes. And finally, we wanted to know about the relative levels of genetic diversity for those 3 different lineages. And so I've generated over the last 3 or 4 years a genetic data set of about 1,600 individuals using a genotyping by sequencing approach. I'm not going to go into many of the methods today, but I'll just give you a brief overview. Genotyping by sequencing is analogous to a dd-rad-seq data set. It produces thousands of different SNPs that we can use to ask these questions. And we primarily use 3 different types of analysis today. So we use principal component analysis, which gives us little insights into individual levels of variation among these lineages. We also use the hierarchical Bayesian clustering analysis that's similar and based on the correlated allele frequency model from structure, which you saw in the previous 2 talks. And then finally, we estimated nucleotide diversity for all the different populations in the state. All right, so the first question is there genetic structure among the lineages? I'm first showing you a principal component analysis. I'm plotting the first 2 principal components on the X and Y axis. Each individual circle represents an individual bighorn sheep. They're color-coded by the 3 different lineages. And what you can see is overall, the 3 subspecies cluster really tightly, and there's great separation between them. So this tells me that the lineages are all very strongly differentiated. Now you'll notice that there are a few, 2 or 3 individuals that appear to be different colors in the different groups. I'm still working out whether or not those are mislabeled individuals or potentially wandering individuals. Still need to dig deeper into that. Also, you'll notice that there's a group of intermediate individuals in the center of the plot that are colored either Rocky Mountain or desert individuals. And this is consistent with desert Rocky hybrid individuals. And so while we thought that there might be some similarity in potential gene flow between Rocky Mountain and California bighorns, we're actually only seeing hybrids between Rocky Mountains and deserts. And so now I'm going to show you the results of those ancestry plots, and I'm just going to show you the pie charts on a map across the state, similar to what you've seen in the previous two talks. And so I'm only showing you the northern portions of the state. For each herd, I'm showing a pie chart filled with the proportion of ancestry from 3 different lineages. And so you can see for most populations, we have pure ancestry for one of the 3 lineages. But there are 2 herds in particular where those hybrid individuals are found. The first one I want to talk about is the white pines, which is a little bit more southern. And this one we knew happened. So there were reports about 20 years ago where individuals moved from this core Rocky group in the Rubies and East Humboldts down south along ridge lines into the White Pine Range, which made hunters that year really excited because these big Rocky individuals showed up in the hunt unit, and also led to very limited hybridization. Really, I'm only picking up hybrids for 1 or 2 individuals. So it doesn't really look like those individuals had a huge impact on shaping the ancestry in the White Pine Range. On the other hand, the herd in the northeast portion of the state, the Badlands, almost every individual in that population is roughly 50-50 desert and Rocky. And this is really surprising to us. We didn't know or have any inkling that this population was a hybrid. And you'll notice it isn't really close to any desert herd out there. So we're not entirely sure where these desert individuals came from. We thought this was going to be just a pure Rocky herd. And so this really warrants more attention. And we really have to come up with some hypotheses on how these desert individuals moved so far. Next, I want to zoom in on only the California populations to see if, to look at genetic structure within this lineage. And so you can see here, I've colored the 2 different ancestries for these herds, like green and yellow. And after examining the translocation history of these herds, these ancestries really relate to the 2 founding sources in British Columbia, both Williams Lake and then the Penticton region. And so this makes a whole lot of sense. There's not any geographic genetic structure within California bighorn sheep. All the genetic structure is related to translocation history. It's also worth noting that there was one pioneering herd in the south here, the Sawabe Range, where bighorn colonized by themselves without translocations. We weren't sure where those individuals came from, but it does look, based on the ancestries, that they came from the neighboring Virginia range to the west. I'm not going to dive into the genetic structure within Rocky populations, just because we only have 2 pure populations. But for desert bighorn, I've previously published a paper in 2019 looking at the genetic structure of these Rocky herds. So I'll just summarize that here. Again, I'm showing you a principal component analysis for 4 source herds, native source herds in Nevada. And we had 1 Great Basin source here in purple, Lone Mountain, and 3 native Mojave Desert source herds in yellow, brown, and blue, which are all very geographically proximate to one another. And you'll see that the first principal component really separates out this Great Basin population strongly from the other Mojave populations. But even for these source populations in Mojave, which are only separated by about 30 kilometers, we still see fine scale genetic structure. And so our conclusions from this were that there was strong structure for these native herds, especially around Las Vegas, but also that there might have been, prior to these translocations and prior to the die-offs, a Great Basin lineage of desert bighorn that wasn't really recognized or thought of before. And this mirrors the analyses done by Mike Buchowski in California, who's also found some evidence for this Great Basin lineage. Looking at the rest of the desert populations in the state, this is a network showing the translocation history of individuals in the state for desert bighorn for the different populations. The size of the line directly relates to the number of individuals moved from one herd to another. You can see the 4 different sources that you saw on the previous slide. And I've looked at this slide for so many hours of my life, but the take-home point after you look at it for some time is that the genetic admixture, the nature of the pie charts, the colors of the pie charts directly relate to the translocation history, the network in the middle of the circle. And so we've lost geographic genetic structure of desert bighorn in Nevada, and it's been replaced by this translocation history. Finally, just thinking about genetic diversity, I have a plot here of the 3 different lineages on the x-axis, again color-coded the same as you've seen before, and then nucleotide diversity on the y-axis. And nucleotide diversity, you'll note, is really small, much smaller than the heterozygosity estimates you might see from, say, a microsatellite data set, because they're not directly comparable. But you'll see here that it doesn't appear that genetic diversity varies across these different lineages. And we had some evidence that it might, but I'll talk about that a little bit later. And so to summarize my results quickly, we have found from this data set that all 3 of these lineages are strongly differentiated from one another. And there's, of course, one small caveat to say that we have only sequenced 2 pure rocky populations. And so, you know, we would feel much more comfortable strongly saying this if we had more of those rocky, pure rocky herds sequenced, especially rocky herds from the native range. Second, we've found hybridization between rocky and desert lineages. This wasn't necessarily what we were expecting. We were expecting potential hybridization between California and Rockies. And we have to think more about how these individuals might be moving around across the landscape. Again, we've found potentially a previously unrecognized Great Basin lineage of the desert bighorn. And so there's only 1 pure herd left in Nevada with this ancestry. And so we have to really think carefully about how we want to manage and protect that ancestry moving forward. It looks like, at least for California and desert bighorn, that genetic structure within lineages is shaped largely, overwhelmingly by translocation history. And so just a reminder again that there are those genetic consequences of translocations, and this is one of them, right? We have an erosion of population genetic structure, geographic genetic structure, and that's something that we need to think about moving forward. And finally, we have relatively similar levels of genetic diversity across these different lineages. So what does this mean for management? In particular, I want to think about management of California bighorns in the future, right? And so there's been a lot of discussion over the past 20 or so years about how we should manage California bighorns, whether or not they're an actual lineage, whether they're different from Rocky Mountain bighorn. And I think the genetic evidence today strongly suggests that they are independent lineages. And despite the morphological study by Weyhausen and Ramey, we might not necessarily want to mix these individuals via translocation. However, when thinking about the translocation history of California bighorn across the Western United States, most of these populations were founded by a handful of regions in British Columbia, and they've undergone several founder effects and bottlenecks. And so they've been previously thought to have fairly low levels of genetic diversity. Further, translocations of Rocky Mountain bighorn into North Dakota led, where it was previously California bighorn, led to higher population recruitments, suggesting that mixing these lineages, especially when trying to match the bighorn to the appropriate environment, might have positive consequences. And so really we need to think about what circumstances we might want to think about mixing these, whether or not we should. And so moving forward, we are trying to develop a genetic management strategy for California bighorn sheep. And this is being led by Clint Epps at Oregon State University, together with myself, and then again, Marjorie Matoke and Tom Parchman at the University of Nevada, Reno, and the Wild Sheep Genomics Working Group. And we're trying to develop a genome-wide and range-wide genetic data set of California and Rocky Mountain bighorn sheep from Nevada up to British Columbia, down the Rockies to Colorado. And we're going to characterize the degree of separation between Rocky Mountain and California bighorn and determine thresholds of genetic diversity where intervention may be warranted, and really try to give managers a solid concrete plan of how to manage California bighorn sheep moving forward. We have several participating jurisdictions that have already provided samples. It looks like we're going to end up with this data set of about 3,000 sequenced individuals. And I'm really hopeful and excited to share what we find at the next meeting in 2 years. And with that, I want to thank funding, University of Nevada, Reno, the Nevada Department of Wildlife, and the Wild Sheep Foundation. And I probably am out of time, but thank you.
All right, thank you, Josh. Great data and great talk. So we have a couple of questions in the Q&A. So I think we have time for both of them. So the first one is from Karina Lemmy, and she's asking why you think there isn't gene flow between California and Rocky bighorns. Is it the landscape or is it behavior limitations?
Yeah, that's a great question. So in Nevada, most of the mountain ranges run from north to south. So if there is going to be natural gene flow, you would expect it to run north and south as opposed to east and west. So that might be something to do with it. There is one California herd with very low levels of Rocky ancestry, less than 10%. And I'm not quite ready to say that there might have been gene flow from Rockies into that California herd yet. It still requires some more investigation, but it is possible that it has happened. And of course, behavior, it's really hard to predict which individuals will be successful in gene flow and will have positive fitness after moving into a new population. Those white pine individuals, they're big rams, big Rocky rams, and we would expect them to outcompete desert rams, but it doesn't really look like they had much of an effect. Whereas in the Badlands herd, it looks like whichever individuals were quite successful at mating. And so there might be a lot of stochasticity in that as well.
Thank you. And I guess we're running just a bit late, but I'll ask you the last question if you have a short answer. So, okay. So Marco Festa-Bianchette says that one issue is local adaptation. So he's wondering if you could use your data maybe over time to help study this and local adaptation.
Absolutely. I think moving forward, that would be, that's the goal. And so in Nevada, we don't necessarily have many herds that have long-term sampling, and hopefully eventually we'll get a data set that could have that. And also in Nevada, we don't have that many native herds. And so it's a little harder to disentangle local adaptation when the translocations are having such a large effect. And so hopefully either by looking at native herds more across the range or having extended sampling, we can really get to that in the future. Thank you.
Great, thank you very much. So we'll be moving on to the last speaker of this session.